Today, I will share a proof of concept framework for topology guided protein design using ESM3. The key idea of this project is to guide a large pre-trained model using non-differentiable topological constraints, specifically mathematical knot type, without having to retrain the model. So just for some background knowledge, the task that we're interested in is designing knotted proteins. Previously, this has mainly been demonstrated experimentally with a more recent study from 2023 using AlphaFold2 and Rosetta-based sequence design to guide the knotted protein backbone design. However, the paper itself was still mostly focused on the experimental and chemical synthesis of the knotted proteins. It's always been challenging to design proteins either experimentally or computationally when we are trying to focus on the more complicated knotted proteins, such as the 5 crossings or 6 crossings. So that's why we picked this task, and due to the challenging aspects and also the biological prospects of it, given that knotted proteins have been demonstrated to improve stability, and they are less likely to degrade. The model that we leveraged is ESM3, which is a mask multimodal transformer model that jointly learned the distributions over sequence, structural, and functional space of proteins. And to enable conditional generation, we use what is called the derivative-free guidance approach, which steer the model to generate proteins of specific mathematical knot types without having to retrain the model. So during each iteration of the generation, we will calculate a reward function for the candidates and bias the model towards our target knot type distribution of the protein. For the reward evaluation, we use what is called the Alexander polynomial, which is a knot invariant, meaning it is able to differentiate different knot types. In the appendix section of the paper, we have a very detailed demonstration of how to compute such a polynomial for the trefoil protein. So one thing to note that even though this invariant is not a complete invariant, it is, however, sufficient for our use case since we're not designing proteins of 10 crossings. We are simply interested in designing a trefoil protein de novo. So just to share some preliminary results, we looked at the typical sequence length of knotted proteins just to have a general idea on whether the sequence length defined in our prompt would even be sufficient for forming entanglements. And we noticed that sequence length did not seem to significantly affect the knot type of proteins. So our demonstration here is a video showing a trefoil protein designed at de novo. Here we introduce a random closure without inducing any artifacts, and we simulated a dynamical relaxation program, and then in the end the protein chain was simplified to a trefoil. This shows that our defined framework is capable of designing a knotted protein de novo, meaning that we did not provide any wild-type template to start with in the prompt. Another task that we got interested in was trying to see if we could generate a protein mutant, and where we put in a wild-type protein that is already knotted. And again, we were able to show that the unknotted variant of a truffle protein turned out to be of sufficient quality. And here the color coding shows the SASA of the protein. So for more details of the project, feel free to check out the GitHub repository, and there's also another linked GitHub repository for detailed instructions on how to check out molecular knots and how to use the KnotProt visualization tool.